Hello, and welcome to your second notes video for Unit 6. Last class, you learned about DNA's structure and how DNA replicates. This class, we're going to learn about DNA's sidekick, RNA, and the adventures these two go on. RNA stands for ribonucleic acid. RNA, like its bigger friend DNA, is single-stranded. It's a very skinny molecule. RNA comes in a couple different forms. There's mRNA, which is like half of DNA. It's one strand. There's tRNA, which looks like a T, and there's rRNA. During today's class, we're going to specifically focus on mRNA and tRNA, the messenger RNA and the transfer RNA. So, RNA, just like DNA, has nitrogen bases on it, except RNA does not have T for thymine. Instead, RNA has U for uracil. C always pairs with G. That's true for DNA or RNA. But since there's no T on RNA, A pairs with U. AU, CG. So, why does DNA need a sidekick? DNA needs a sidekick because it goes through the large process of protein synthesis. When we learned about photosynthesis, we learned that synthesis means to build. So when we're doing protein synthesis, we are building protein. It all starts with DNA. Let's ask this. Who remembers from Unit 2 macromolecules? What macromolecule are you going to get if you're eating steak? Steak is a great source of protein. Protein builds muscles, and steak is cow muscle. So, if you said proteins, you were correct. We know that proteins make up everything in our bodies. Our skin, our muscles, our nails, even the enzymes that help us make protein are made of protein. So, how are proteins made? Today, we are going to be talking about the story of protein synthesis and answering some questions along the way. Once upon a time, our body needed to make protein for our muscles. This process is called transcription. Our DNA got to work and began writing a recipe to make protein. The recipe is copied onto messenger RNA. The mRNA took the message and said goodbye to DNA and left the nucleus and began his journey. DNA cannot leave the nucleus because DNA is too large. So it asks its friend messenger RNA, its skinny single friend, to come into the nucleus, get the message from him, and take it to where it needs to go. This is why I call mRNA DNA's sidekick. The process of getting that message from DNA and putting it on mRNA is called transcription. DNA copies its message and transcribes it or writes it onto mRNA. How it does this is mRNA comes into the DNA and looks at the letters on DNA. It takes the matching letter message and brings it out of the nucleus. So, here's where you can see this happening in a cell. Inside the nucleus, we have DNA. DNA opens up so that mRNA can make a message from it. Once you have mRNA, it's copied, it will then leave the nucleus. So, in mRNA, which base is substituted with the letter U? What this is really asking is which letter is on DNA, but not in mRNA. If you said the letter T, you'd be correct. There is no thymine on mRNA, but there is on DNA. So if you see here, instead of A pairing with T, A pairs with U. T is substituted for U. Where does transcription happen? Hint, think, where does DNA live inside of the cell? If you said nucleus, you'd be correct. The nucleus stores DNA. DNA is much too large to leave the nucleus because it's double-stranded. DNA does have the information needed to make protein. But because it can't leave the nucleus, it gives this information to mRNA. It writes down or transcribes a message to mRNA to get the ingredients. Let's practice. Here we have DNA that has been opened up. mRNA has come in at the bottom to get its message from DNA. Write the message from DNA onto mRNA. What this really means is match your base pairs. If there's a G, you should write a C on mRNA because G and C pair together. If there's a C on DNA, you should write a G on mRNA because C and G pair together. Here's where it gets a little bit tricky. A normally pairs with T, but there is no T on mRNA. So what letter are you going to put? If you said U, you would be correct. Up here at the top, we have a T for DNA. Which letter does T always pair with? A, correct. Keep going to finish the strand. Now that you've transcribed your message onto mRNA, it's time to start the next step in the process of making protein. This next step is called translation. Now that you have this message, it's in a bunch of random letters. We need to translate it into something it can understand. So mRNA leaves the nucleus with this message that's not in a language it can understand and goes into the cytoplasm. Once it's in the cytoplasm, it calls up its friend, translator RNA. Translator RNA is going to help translate the message into something that the cell can understand. Think about if you are bilingual. You might have gotten a message in Spanish that you need to translate for your friend who doesn't speak Spanish. That's what's happening here. mRNA does not speak in the base pairs. So tRNA comes in to translate the base pairs into something that the cell can understand. This all happens at the ribosome. So this is kind of where they meet up to do the translating, just like you might meet up during Lancer time to do homework with a friend. So, how do mRNA and tRNA do the translating? mRNA feeds the message into the ribosome, where tRNA is stationed to translate. tRNA has 3 letters on it. We call this a codon. tRNA finds the matching 3 letters to that codon and translates it into an amino acid. If you can think back to Unit 2 when we talked about macromolecules, amino acids are the monomers or the building blocks of protein. So as it starts translating 3 at a time, 1 codon at a time, it stacks these amino acids on top of each other to make a chain of amino acids, which is a protein. The translator RNA will continue to translate until it reaches a stop codon, which tells it to stop translating. That's when the protein is complete. So, finally, the protein is sent to other parts of the body where it can be used to make muscles, build immune system, or grow your hair and nails. Where does translation happen? We know translation happens with mRNA and tRNA because T stands for translate. But where do these two friends meet up? If you said the ribosome, you would be correct. Translator RNA and messenger RNA meet up at the ribosome. What does letter A represent? What is the single-stranded sidekick of DNA? If you said mRNA, you would be correct. mRNA contains the message needed. Now, who's doing the translating? What is letter B pointing to? If you said tRNA, you would be correct. tRNA stands for translate. Not only that, but tRNA actually looks like the letter T, so it really helps you remember what it does. So, there are 2 steps to protein synthesis. The first step, transcription, involves DNA, who has the information, and mRNA, who's getting the message. What's happening here is DNA is giving the message to mRNA. This all happens in the nucleus because that's where DNA lives. After transcription, mRNA leaves the nucleus and it calls up its friend tRNA, so to do translation. Translation involves messenger RNA with the message and translator RNA who's doing the translating. They meet up at the ribosome to translate the codons into proteins. That's all for today's notes.